So in your body, you have a few different overarching types of cells. You've got, for one, you've got your cells that are actively able to divide. So some examples would be the epithelial cells that make up your skin, and you've got your fibroblast cells that make up the scaffolding of most of your organs, like say your kidneys and your liver, and even the endothelial cells that line the insides of your blood vessels. So these cells, right, and their precursors, they can all divide by mitosis, and that's to help replenish and regenerate the tissues that they're a part of, right? And actually, we'll include some of our stem cells in here too, the cells that these other cells initially come from. And we'll call this whole set of cells here mitotic cells. They undergo mitosis. So we also have another group of cells in our bodies that we refer to as non-mitotic cells, or actually post-mitotic cells. That's a more accepted name. And you can probably guess why. They don't undergo mitosis. They're incapable of proliferation. And you might sensibly be thinking that, well, hey, that means that they can't repair and regenerate like our mitotic cells over here. And you'd be on the right track. They certainly have a limited ability to repair or regenerate the tissues that they're a part of. And sorry, some examples would be, say, neurons, either within the brain or elsewhere in your nervous system, and heart muscle cells, right? These are neurons and heart muscle cells. These are both examples of post-mitotic cells. But yeah, you're on the right track. This tissue doesn't regenerate by mitosis of these cells, like what happens over on this side here. This tissue over here regenerates only very slowly, and only by asking some special tissue-specific stem cells to help them out. But it's a slow process of repair in post-mitotic tissues, so to speak. So here's the thing though, here's the problem with mitotic division. So what the cells on this side do. So with mitosis comes DNA replication, because in order to create new daughter cells, you have to copy your DNA to pass on, right? But DNA replication in basically every mitotic cell, besides our embryonic stem cells, has this interesting little caveat. So let me show you what I mean here. So here's our cell ready to start mitosis, right? So it wants to copy its DNA to pass a full set on to each of its daughter cells. So here's our DNA. Remember, in eukaryotes like us humans, we have nice linear strands of DNA, and we have lots of them, as opposed to our prokaryotic bacteria friends who have circular DNA. So here's our linear DNA, and actually at the ends of our DNA strands, right, on both sides, we have these little caps, sort of like the little plastic tips on the ends of your shoelaces. And just like how the little plastic tips protect your shoelaces from fraying or getting worn out, our little telomeres, that's what these tips are called, our telomeres prevent damage from happening to our DNA during the copying process. So to clarify, these telomeres at the ends here don't actually code for any proteins. They don't have any genes on them. All the important DNA is in this section here, this middle section. So I said that there's potential DNA damage during the copying process. What do I mean by that? Well, the machinery that copies our DNA polymerase system, it kind of works in a funny way. It doesn't actually copy our DNA all the way to the end. So what we see is that each time DNA is copied, you lose a little bit of telomere here. So every time the DNA gets copied, you lose a little bit more and a little bit more and a little bit more of the end. So lucky for us, instead of having really important coding DNA at the end of our chromosomes, we've got our telomeres. So again, it's the telomeres that end up becoming slightly shorter at the end of each DNA replication cycle. And it turns out that a cell can undergo about 60 or 70 or so cell divisions before the telomeres get too short, and then all this important coding DNA here starts to become at risk of damage from further replications. But not to worry, we have a contingency plan built in. So once the telomeres get too short, the cell initiates a DNA damage response. So it sort of rings these internal alarm bells, and it sort of gives up its ability to divide, because it really wants to prevent any more shortening of this chromosome that would happen in further rounds of replication, right? So it loses its ability to divide. And by doing that, it has become what's called a senescent cell. And before I explain what exactly that is, there's just two things I want to point out about this. So the first thing is that if a cell reaches senescence because its telomeres have become too short, we say it's reached replicative senescence. And the second thing is that the number of times a cell can divide before reaching senescence, replicative senescence, is called its Hayflick limit. And that's named after the scientist who figured that out. And again, it's generally around about 60 or so divisions. So now that we've gotten all that out of the way, what exactly is senescence? Well, basically it's a change in a cell's state from a happy, active, mitotic cell, right, i.e. one that divides on a regular basis, to one that's non-dividing and possibly less happy. I'll have to ask a senescent cell how it's feeling one of these days. But it undergoes all these other changes as well. Like it starts expressing all sorts of genes that it didn't before, and it starts to look a little bit different, and it starts to respond a bit differently to the cells around it. But the important thing is that while it's still contributing to the structure of your body, it's still hanging around in your tissues, it's not allowed to replicate anymore. And kind of the key with all roads to cellular senescence, be it through reaching your Hayflick limit as we just saw, or through other ways, which I'll mention in a second, the key is that cells transform into a senescent state to prevent impending DNA damage from happening and causing maybe cancer or a tumor, or possibly being passed on to daughter cells. So the cell's ability to divide gets turned off. And other causes up here, well, essentially anything that threatens the integrity of your DNA. So maybe if you had telomeres that just don't work properly, or maybe you sustained some DNA damage through a mutation of some type, or maybe some toxin damaged your DNA, any of these could really just as easily result in your cell becoming senescent, non-dividing. So is there actually a problem with senescent cells? Are we happy about them? Are we upset with them? What's the deal here? Well, we're not really sure yet, but conceptually an easy way I like to think about them is that, well, on one hand, they help prevent tumors or cancers from happening because they're not allowed to divide anymore. But on the other hand, as we get older and our tissues have more and more senescent cells in them, the tissue can't really repair itself as well as it used to because there's fewer actively mitosing cells around, right? And there's actually also some evidence linking senescent cells to age-related diseases like cataracts. But this is a pretty active area of research. So for now, we'll just have to keep an eye on these cells to see what new research comes up with for us. Now, let me zoom out a bit here to show you something else. So here's our mitotic cells, right, that by definition are the only ones that can undergo replicative senescence. And over here are our post-mitotic cells. And these guys can't reach replicative senescence, right, because they don't replicate, but they can be induced to become senescent if their DNA is at risk of becoming damaged in basically just the same way as I mentioned earlier on, DNA damage from some source. And the last thing, I just want to draw out a graph here to represent our different types of cells and their tendencies with division. I think this will really help us to get the picture here. So here's our graph. Here's our y-axis here, and here's our x-axis. And on the y-axis, I want to put cell division capacity, okay, so capacity of this cell to divide. And on the x-axis here, I'll put number of doublings, which is essentially how many times the cell has divided already. And this will all sort of make sense in a second here. So our somatic cells, our regular body cells that are able to mitose, to undergo mitosis, they start out, say up here, right, with a pretty high cell division capacity. They're able to divide a lot. And as they continue to divide and divide and divide, they sort of move this way. Their curve sort of curves down this way as the number of doublings that they've done increases, right? So you can see the more doublings they've done, the lesser their cell division capacity, right? And that's because their telomeres, with each doubling, their telomeres are getting shorter. So their capacity to divide is getting lower with the increased number of doublings, okay? But we have another type of cell, right? We have, say, some of our stem cells. They start off up here, right? Let's say they start off at the same place. They have a really high cell division capacity. But as they divide, right, their line, their curve just stays really high, right? Their cell division capacity, regardless of the number of doublings, regardless of the number of times they've divided, their cell division capacity stays really high, right? So why would that be? Why might that be? Why would they be so different from our somatic cells? Well, it turns out that they express a special enzyme called telomerase. And what telomerase does is every time one of these cells from our stem cell curve here every time it undergoes replication and loses a bit of telomere, telomerase, the enzyme, sort of springs into action and adds back that little bit of telomere, right? So that means that these cells are never going to reach a Hayflick limit, right? They're just going to have the ability to replicate as many times as they want. And that's because they express telomerase. And the very last thing I'll say is that sometimes your somatic cells, right, they can develop a mutation in their DNA that causes them to start expressing telomerase. So now they have the enzyme telomerase present within the cell, right? So now they jump off our curve here, our regular somatic cell curve here, because they get the ability to replicate as many times as they want. They essentially escape senescence, and this is one of the ways that cancer occurs, because now you have a group of cells that can go on and multiply out of control.